Welcome to our presentation on a modular bioimage analysis workflow for analyzing cell shape transitions. This workflow was originally developed as a part of reproducibility assessment of AmoePy, a software platform designed to quantify cell shape dynamics during migration. The workflow is modular and can be readily adapted to many other cell shape analysis applications. Today, we will walk through the complete workflow from image segmentation, mask refinement, contour extraction, and spatiotemporal analysis. As cells continuously change their morphology over time, the first goal of this workflow is to accurately define the cell boundaries at each time point. After generating cell masks, the contour is parameterized using virtual markers distributed along the cell parameter from 0 to 2 pi. These markers along the cell parameters are then linked across consecutive frames, allowing us to track local dispersion changes over time. Based on the movement of these markers, we can quantify local dispersion along the cell boundary. In the resulting kymographs, warm color indicates protrusive activity, whereas cool colors represent retraction, providing a spatial temporal view of cell shape dynamics. This workflow consists of 4 major modules. First, we use elastic pixel classification to identify cells and generate initial segmentation masks using Fiji. Since row segmentation often contains imperfection, the second module focuses on mask refinement, including the removal of irrelative objects, filling internal holes, and filtering out debris based on object size. Once masks are obtained, the third module extracts coordinates from the cell masks and converts them into a text file containing marker positions for every time point. These contours serve as the primary input for the final module, where AmoePy performs a spatiotemporal analysis of cell shape transitions. Now let's watch a short demonstration provided by Esteban, showing the first step of this workflow, elastic-based segmentation. This video illustrates how to use a well-trained classifier to identify cell regions and generate the initial segmentation masks.
Hi, I'm going to show here how to use the supplemental materials included in the reproducibility assessment of cell shape transition analysis in AmoePy paper. First off, I'm going to show you how to use the elastic project file that is included in the elastic folder within the paper's repo. All we need to do to open it is drag and drop it into the elastic software. This project contains a pixel classifier that has been already trained on several sample images of amoebas in order to detect or to classify the pixels as belonging to the background or to the cells. The objective here is to detect the cells and increase their contrast with the background. So training is already done, so you don't have to do anything. Similarly, in the prediction export, this is already set to export the probabilities within the same folder where the images are in a subfolder that is going to be called elastic and the result type that is probabilities. So nothing to do here. All we need to do then is go to batch processing and select the raw data files. In this case here, I have my original amoeba images. So I'm going to select all the images in the folder, click open and click on process all files. This is going to take some time, but you already see that Elastic created the output folder here in the same folder where I have my raw images. So now that the process finished, I can see that I have all the output files that Elastic produced. We can open this folder in ImageJ to see the output of the Elastic Pixel classifier. So the next step is to use one of these ImageJ scripts included in the supplemental material to process this output of Elastic to create a binary mask. In order to open it, we just drag and drop it into Fiji. And here, once again, you don't need to edit anything in the code. You can just hit run and select the folder where the output of Elastic is stored. In this case, it's here in this Elastic probabilities folder. I can just open it, it will process it, and as you can see, creates in the same parent folder a post-processed new directory that we can open. And if I get rid of this, you can see now that this is a binarized version of the probability outputted by Elastic. So now this is the raw data that we are going to input in AmoePy to perform the tracking of the contour over time.
At the previous step, we have obtained the initial mask images. However, segmentation output often contain imperfections that can interfere the downstream analysis. In the next demonstration, Angie will show how these masks can be semi-automatically refined by removing irrelevant objects, filling holes, and cleaning artificial debris to produce analysis-ready masks.
Now I'm going to show how to remove non-relevant objects from images before contour extraction. The first step is loading the images. Here, we use import image sequence function in Fiji to load our images as an image stack. As you can see, this image has 3 problems that need to be fixed. First, there are unwanted cells outside our target cell. Second, there are holes inside the target cell. Third, there is a cell debris that should not be included in the analysis. All of this need to be removed before contour extraction. To remove unwanted cells, we can use the trajectory of the target cell as a mask. First, use the Z projection function under the stacks menu to generate a projection image showing the cell trajectory. Next, select trajectory, use the Match 1 tool, and add it to the RI manager. Then, apply this RI to the entire stack and use clear outside function to remove objectives outside the trajectory region. For the holes inside the cell, we first convert the stack into a binary mask. And then use the fill hole function to remove them. At this point, only the cell debris remains. To remove the debris, we can use the size filter in Analyze Particles. Before setting the filter, we need to determine the area of the largest debris object. First, make sure that no RI is selected on the mask stacks. Then, open Analyze Particle. Set the size ranges from 0 to infinity. Choose Show Nothing and enable Exclude on Edges and Add to Manager. Run the analysis on the entire stack. After all RI has been added to the RI manager, click Measure to obtain their area measurements. Next, sort the area column in the result table, and look for a clear gap between the large and small objects. In this example, row 551 appears to be a largest debris candidate. To verify this, we need to inspect the corresponding RI. The RI IDs are listed in the label column of the result table. Simply select the corresponding RI in the RI Manager to display it on the image. And from the image, we can confirm that the row 551 is indeed a debris object. While the row 552 corresponding to the target cell. Now, we know that the largest debris area is 151 pixels. Let's run analyze particle one more time. This time, set the size ranges from 152 to infinity and choose the show masks. After running the analysis, our debris objects smaller than the target cell will be removed. Finally, we obtain a clean image stack that's ready for contour extraction.
After obtaining clean masks, the next step is contour extraction. This video demonstrates how to use the provided Fiji macro to extract coordinates from each cell mask. The extracted coordinates recording the position of coordinates markers at every time point, and serves as the direct input for subsequent analysis in AmoePy.
Hi, this video demonstrates how to extract contour coordinates from a binary image stack for AmoePy using our Fiji macro. First, drag and drop the macro file into Fiji to open it. Next, open your binary image stack. Once everything is ready, click the Run button. A setup dialog will appear. The first option is the destination folder for the output file. And the macro will automatically select the current active image. But you can still verify or change it using the drop-down menu. So, the sample points specify how many points will be extracted from each contour. The default value is 400. And start time defines the timestamps of the first image in the stack. Frame interval is the time between consecutive image frames. The macro will calculate the timestamp for each frame based on this value. The last parameter is pixel per milliliter. This scaling factor is used to convert contour coordinates from pixels to micrometer in the export file. After confirming all settings, click OK to start the analysis. When the processing is complete, a log message will appear indicating the contour coordinates have been successfully exported.
Step 4 focused on spatiotemporal analysis within the AmoePy environment. The process begins by importing the cell contour file we just got and performing the outlined alignment through minimizing mean-square displacement, which will generate a new resolved file in the same folder. The user can visualize the aligned cell track by clicking the button here, Cell Track Plot, to get the plots on the right hand. We then go to marker connection by activating the button here, Compute Chemograph Files. The default parameter of these parameters are adapted to the analysis, and the output path can be specified within this column. Then start the analysis. It can take a while depending on the computation power and also the frame size of your dataset. In this case, it costs about 14 minutes to complete the whole analysis. Finally, we evaluate the analysis generated by AmoePy. The workflow also generates a variety of output, including motion fact sheets and chemograph plots. Together, these results provide a robust framework for quantifying the spatiotemporal dynamics of cell shape transition during cell migration.